Good morning, good evening, good afternoon, good day, everybody. So thanks for joining the second day of the workshop about dangerous liaisons. For those who did not attend the meeting yesterday, I just want to highlight that this is an event to celebrate our 20th anniversary of the establishment of Hong Kong HKU-Pasteur Research Pole in Hong Kong. And because of COVID, we really want to before COVID, we really wanted to bring you over here to actually join the celebration. But because of COVID, and so now we try to break down these celebration event in multiple smaller pieces, and this is a part of the celebrations. Right, yesterday, we had a major emphasis about coronavirus and their host. Right, we had experts talking about coronavirus transmissions, either in human or in animal. And then we also talk about the ecology of this virus and the host adaptations, etc. We also had a very interesting discussion about this group of pathogens, like how does it emerge, or did it emerge into human, etc., etc. But I want to highlight that nature may have already warned us about this pandemic well before, right? Maybe dating back to 2003, when the first epidemic of SARS occurred. And technically speaking, SARS coronavirus and SARS coronavirus 2 virus are classified as the same virus species. So they are very similar, and they have been circulating in animal all the time. So we just don't know when it will hit us again, right? And of course, in the last 13 years or 17 years, we have done a lot to understand the ecology of these emerging viruses. But I think we are not doing enough to prevent or even stop the pandemic, right? And yesterday, you may also heard about other important emerging pathogens or the emerging pathogens like Zika in South America, avian influenza virus in Europe, in Asia, and in fact, H5N8 has been actually endemic in a lot of countries. So we are still having a constant threat of these emerging and the emerging viruses. Okay, so and this really highlight the needs of international collaborations and the needs of international research network that is mandated to address these health threats. Right, just now in African countries, we are having 2 countries having Ebola outbreak at the moment. So we are under constant threat. And this is why we have to set up this particular research network at Hong Kong. Located in 25 countries on 5 continents, the HKU-Pasteur International Network includes 32 institutions, which are united by shared values and missions for the benefit of populations. Present in numerous endemic areas, the international network has, time to time again, demonstrated its major role as a sentinel for emerging infectious disease. The network holds a health and epidemiological surveillance role, which is essential for responding to epidemics, understanding the environment and epidemiological mechanisms that lead to the development of endemics, and detecting the emergency of or the emergency of infectious disease. Right, today, now we are going to change the topic slightly. We have a panel of people who are expert on entomologists, epidemiologists, and also anthropologists, right? And then they are going to focus on the necessity of enhancing virus surveillance. In particular, we are very pleased to have members from other Pasteur network or institutions from different countries, so to facilitate the discussions. And then we also have a special focus on anthropology, and we have an anthropological perspective, which will also be brought to the discussions, highlighting how cross-discipline can help to improve, to combat infectious disease. Right, but before the panel discussions, we're going to have a keynote lectures. And yesterday, we have been talking about surveillance, and we actually learned quite a lot of surveillance. And one key question was, with this existing information, can we better predict or identify pathogens that are found in animals which have a pandemic threat to us? right? And we have our keynote speaker, Dr. Marco Venusi. So he's going to talk about these particular topics, how we can better predict or identify pathogens that may have a pandemic threat or health threat to humans. Right, Dr. Marco Venusi, he was trained as a biologist in microbiology and immunology at McGill University. So he has a first bachelor degree in Canada, and after that, he moved to University of Paris and doing fundamental biology as his master degree. After that, he joined Sylvia Vanderveer's group in Paris again to study influenza viruses. Right. After that, Marco went to California, and he joined Ralph Adino's lab at University of California in San Francisco. So to study virus evolutions. And there are a lot of exciting, I read a lot of his paper in his lab. There was a lot of exciting discovery from his postdoctoral studies. And after that, he moved back to Paris and established his own group. Now he's running a lab concerning virus population and pathogenesis. Right. Today, he's going to present his work about arboviruses. And his topic is experimental system to study the cross-species transmission and within-species adaptation of mosquito-borne flavivirus and avivirus. Right, without further ado, may I pass the platform to Marco? But before that, I forgot to mention that right now, Marco have a severe laryngitis. So he cannot actually speak. So now this is him. So now, but he can do hand sign, but he cannot speak. So now I'm going to read the script on behalf of him. Yep, this is nature. So all are susceptible, all humans are susceptible to be infected, right?
Bacterial infection.
Yes, exactly. This is a bacterial infection. But then again, this is still an infection. So right now, so I'm going to read the script that he is kindly prepared for us just now. So I will try to read it slowly. Okay, slide 1. Good afternoon, everyone. I'm sorry for not being able to present my work to you as planned. I hope that this alternative will give you some idea of the newest finding we have been working on. Today's talk will focus on our newest work addressing whether defective viral genomes derived from arboviruses can be used as antivirals and vector control strategies. Second slide. As many of you know, we study viruses as populations or quiescent species. To put it simple, instead of a single genome like the number shown here, we consider that the virus make numerous mutations or variants at each round of replications. Slide 4. As these variants grow in number and diversity. Slide 5. With each round of replications. Slide 6. So that even after a few rounds of replication in a single sample, you don't have just one virus, but a cloud of variant or quiescent species. For the first 10 years, my lab has focused on looking for which of these variants have an adaptive advantage, which point mutation are the best. But in reality, there are other types of variants, such as genomes, that are not full-length copies, that have deletions. They are commonly called defective viral genome, or DVGs. Next slide. This variant were first discovered by von Manners in the late 1940s. He severely passaged influenza virus and observed sharp drops in infectious virus titers. These von Manners particles interfered with wild-type virus replications. Later, Huang and Baltimore gave the name defective interfering particle to these variants that could be packaged into virion, but were missing part of their gene sequence. For decades, DVGs were a problem to avoid in cell culture and considered an accident of lab conditions. But now we ask, can these DVGs be useful to us? Next slide. To answer these questions, we set out to develop new ways to identify defective genomes compared to the classic method used in the past. Here is an example of a classic flavivirus DVG. In the past, scientists passaged virus at high multiplicity of infections over and over, and then used RT-PCR assay to pull out the DVG. However, these methods are biased and usually pull out only the smallest DVG with the biggest deletions, such as the DVG shown here, which has deleted all of the protein and coding sequence and keeps only the 5' and 3' ends, compared to the normal genome shown about it. Next slide. We used an unbiased approach where we grow virus at high and low multiplicity of infections, and then deep sequence and use computational analysis. This method easily finds deletions, rearrangements, insertions, and copybacks. It can find DVGs of any frequency, not just the most abundant ones. Instead of finding only one DVGs, like classic approaches, it finds hundreds to thousands of different DVGs in each sample. And it scores each DVG for fitness to determine which DVGs may be better at competing with wild-type virus and acting like parasites. Next slide. The output of our pipelines includes a deletions heat map like this one. Here, the x-axis represents the genome nucleotide coordinates, from the first nucleotide to the last one. Each row of the y-axis represents the cell passage number of a series of passaged samples. The red coloring indicates that this particular nucleotide is deleted at high frequency. This particular heat map of Zika virus passaged in viral cells shows that between passage 9 and 12, there is a very clear preference to delete over 2,000 nucleotides at the beginning of the genome. Next slide. It's important to state that we cannot simply rely on biological triplicates like many of us are used to. In evolutionary experiment, 3 is not enough. Here, we found that at least 12 replicates were required to capture all of the DVG's diversities that was compiled together in the previous slide. Next slide. Another important reminder, if you try this at home, is that each cell type can give different DVG's profiles. About Zika viruses passaged in vivo cells on the left, and in BHK cells on the right. Below, 2 strains of chikungunya virus, the Caribbean strains on the left, and the Indian Ocean Island strain on the right. Not only do the 4 cell types yield different results for each strain, but each virus strain had unique DVGs too. Next slide. When we began our studies, only 1 DVG for influenza virus had been very well studied by Demont and Eastern groups, proven to work as an antiviral in cell culture and in animals, and was making its way toward clinical trials. We wanted to address whether such DVGs existed for arboviruses as well. So in the next few slides, we'll describe our most recent publications, published this month in PLOS Pathogens. It is the main work of Lara Levin, PhD student shown here, and a collaboration with Carla Shashi labs in Institute of Pasteur. This is the chikungunya virus story. First, we passage chikungunya virus in 2 mammalian cells and 2 mosquito cell lines. In the graph, the x-axis indicates where along the genome the deletions begins. On the y-axis is where the deletions end. Each point represents a different DVG. We found 4 distinct clusters of deletions that were common to all cell types and were found in both strains of chikungunya virus. From this computational analysis, we make DVGs corresponding to all of these clusters. We then test them for their ability to interfere with wild-type replications in cell culture. Here are examples of DVG antiviral activity in mammalian cells in the top panel and mosquito cell in the bottom panel. Here you can see that most of the DVGs predicted to compete with wild-type virus are indeed able to interfere with virus replications. When these DVGs are given at a 10-fold excess ratio, they reduce virus titer by several orders of magnitude, and not due to interferon activations, but to direct competitions for cellular resources. We then test whether DVGs could have broad antiviral activities. Here, we used DVGs divided from the Caribbean strain of chikungunya virus, and we test them against closely related alphaviruses, circled in red in this phylogenetic tree, compared to the most distantly related SAMB virus, circled here in blue. Our results show that most of these DVGs can inhibit the closely related Indian Ocean Island strain of chikungunya, but also its closely cousin, Oyan virus, and some even inhibit malarial and a more distant relative, but none of them could inhibit SAMBs. We then move in vivo. We injected mosquitoes with DVG's RNA that we transcribed in vitro. After 2 days, we fed mosquitoes a blood meal containing chikungunya virus. After 5 days, we dissect the midgut and carcass to measure virus infections and disseminations. The results show that while infections still occur in the midgut, the viral titers and the percentage of mosquito with disseminated virus are lower. These exciting results suggested to us that this may impact transmission of virus. Could this be a new way to control virus spread? For that, we shift our attentions to the last few slides, where I described our recent paper that will soon be in press, led by our postdoc Veronica, who has just left our lab to run her own group at Janssen Pharmaceuticals. Using the same pipelines, we identified a whole series of different deletion hotspots that correspond to DVGs of predicted high fitness and ability to compete with wild type. Among these, we focused on what we call here DVG-A, which had the highest predicted fitness. Compared to the Zika virus genome above, our DVG was missing the first few thousand nucleotides at the beginning of the genome that cover most of the structural proteins and a pieces of non-structural protein, NS1. These DVGs resembles a replicon, which are genomes that retain their ability to replicate themselves and are only missing packaging protein. Okay, and then next, we confirmed that DVG-A is not a replicon. A true replicon, shown in black bars on the left side of the figure, can replicate by itself, even when the virus is not there. Here, you see the increasing replications over the 72 hours period, even when the virus is absent. Our DVG-A, shown in the dark blue, shows no replication when virus is not there. When virus is added, then DVG-A wakes up and acts like a replicon. This is a very unique feature which may explain why the DVG scores with such high fitness. Next, we developed a packaging system to create virus-like particles, or VLP, that could be used to deliver our DVG RNA in vivo. To do this, we provide the Zika virus structural proteins and NS1 in trans. This system packages our DVGs as efficiently as it does a normal replicon. We obtain up to 1 million VLPs per mil. We then treat mice with VLPs containing DVG-A and infect them with wild-type Zika virus. We found that virus titer in blood was significantly reduced between 2 to 6 days after infections. Importantly, Virus load was 1 to 2 orders of magnitude lower in the critical target organs for Zika infections, such as the ovary and in the brain. Slide 27. Finally, we returned to the mosquito studies. First, we injected DVG RNA into mosquitoes and observed that RNA persists. It remains detectable even after 10 days. In the paper, we showed that this DVG RNA is converted into a DNA form by the mechanism discovered by the Senel lab. That this DNA form continues to transcribe DVG RNA long after the first injections. Finally, we fed mosquitoes blood containing Zika viruses and then measured the effect of DVG treatment. As with chikungunya virus, all mosquitoes had active virus replication in the midguts. But mosquitoes that were treated with DVG, there was a significant reductions of early and late disseminations, as measured by Zika titers in the carcass and in the head of the dissected mosquitoes. Most importantly, we salivated mosquitoes to address transmissions. In this case, mosquitoes treated with DVG RNA had no detectable viruses in the saliva. Virus transmission was therefore blocked. Okay, to conclude, we described a new experimental and computational method to identify DVG antiviral candidates with high successful rates. We showed that chikungunya virus and Zika virus DVGs can inhibit virus in mammalian and mosquito cells. We showed that DVGs from one virus can inhibit closely related viruses. We showed that DVGs can be used to inhibit arbovirus disease in mammals. We showed that DVGs expressed in mosquito inhibit dissemination and block spread, which proved the concept of vector control strategies. And then in the next 2 new years, we are now moving to work phase 1 clinical safety trial for Zika DVG. Finally, thank you all for trying to follow this clock. With that, I would like to thank my wonderful team that I show here. Together, we are a wonderful circus of scientists that are not afraid to show our unique mutations and variants. Thank you. Okay, so Marco, thanks for your talk. Right, okay, so now we move on to Q&A. For those who have questions, you can either key into the chat box, or for those who are in persons, you may want to raise your questions right here.
Yes, I have the microphone.
Okay.
And there is a moment of silence. I will ask Marco, and he can maybe type his reply. Where are you trying to do this clinical trial for Zika at the moment for safety, and what type of regimen of are you planning at the beginning to verify the safety of these DVGs as possible antivirals? Can you give us a little bit more of details of your strategy at the moment?
It will take a second, or a minute. Yeah, he's doing the typing. Right, so he had replied, regarding to the safety trial, the plan is first to do two studies. And then once we are sure that the DVG is not a replicon and is inert, test in a small set of humans to determine whether it's induced innate immune response. Yes, it's very important to make sure that it's safe to be used, right? And right now, there's no plan yet to test whether it's truly antiviral. But then with the safety trial, then I'm sure Marco will consider the next. Right, and then there's questions about the DVGs generated in cell culture and whether it's actually similar to the DVG found in mosquito, right? So Marco is typing it. So, okay, so we see many more DVGs in cell culture. But in the chikungunya paper, we also found the same DVG in mosquito. So basically, they are not identical, but they have overlaps. One difference is mosquito, we found many more rearranged DVGs. And these were more powerful at controlling infection in mosquito. Well, interesting. So are you saying that a mosquito actually tried to pose a more potent selection pressures to the virus, and so that the virus actually is struggling, and then they have to do more? We believe that, this is the answer for Marco. So we believe that the virus used these kind of DVGs to allow the mosquito immune RNAi response to maintain virus persistency. Okay. In a way, because of these DVGs, these viruses last longer and can be transmitted. Great. Yeah, very interesting. But when we perturb the ratio of the DVGs, the mosquito no longer can maintain persistence and die. Right, okay, so maybe you need more mosquito to put in your experiment. Yeah, but it's interesting. So, I mean, any other questions in the audience? So we have a question from Sophie.
Thank you, Marco. This is a really cool concept. With the different cell lines, how you're seeing different pathways of what's actually being selected, what does it tell you about the host sort of infection pathway in those particular cells? Because they're all susceptible cell lines that you're using, but you're getting different selection pressure on those viruses. So have you picked up any unique host proteins or pathways through the DVG mapping?
Yeah, he's answering. Yeah, sorry about I didn't mention that. He's answering. He's busy. You can see that he's typing. Right, okay. So far, we haven't been able to find any biological reason for the difference. It isn't whether the cell type has interferons or not. It does not seem to be related to the temperature, right? So basically, he did not have the answer. He does not have an answer yet, right? So that's good. That means more research questions, right? So any other questions? Okay, if not, then thanks, Marco. I mean, I'm sorry about your sore throat, and I hope you will get better soon, right? I mean, sure, next time we will invite you again to give a talk using your voice, right? Okay, so now, yep.
Most people are thankful that I can't speak.
Right, okay. So thank you very much. So now may I move on? And then can I invite Dr. Chris Marks to introduce the next speaker?
So thank you very much, everyone. So, and welcome to join our session here. So in the coming sessions, so we have invited several scientists from their own expertise area to share the view of their understanding on viruses. So today we have 4 speakers that will give the short talk and on their own research area. Unfortunately, Dr. Amadou Salah cannot join us because of the current Ebola outbreak in Africa. So may I introduce the first speaker, Dr. Ferric Kick, who is an anthropologist. So he is a director of research at the Laboratory of Social Anthropology of the French National Center for Scientific Research. He is also a fellow of the Canadian Institute for Advanced Research. He is generally interested to understand how microbial act as a science of environmental changes and how non-human beings act as sentinels when they carry these microbials. For example, one of his research focuses on the history of social anthropology and contemporary political questions raised by avian influenza. Without further delays, Franz Ferret.
Hello, everyone. Thanks, Chris, for the introduction. So this talk is a kind of introduction to the panel, and I share my screen. So the panel is on the surveillance of emerging viruses. We will move from coronaviruses that we saw yesterday to all kinds of viruses, including arthropod-borne viruses. Let me start by a quotation from my colleague Vanessa Monceron, who makes a distinction between 2 forms of surveillance, recording and monitoring. So recording is collecting data on a natural site to preserve biodiversity. And that's what bird watchers do, for example, in Hong Kong. They're very active to produce data on the bird species. And the monitoring is a vigilant attention to early warning signals in a hotspot of emerging threats to anticipate a crisis. And that's what veterinarians do, for example, in Fujian, when they collect samples that are then analyzed in the laboratories in Shantou or Hong Kong. And the question we saw yesterday of which samples are interesting to study and how we can focus on some samples to make them meaningful really involves a shift from one mode of surveillance to the other. But also it raises the question of how people who do surveillance on a regular basis, just for the sake of knowledge on a natural site, can be included in the monitoring to anticipate future crises. And so to think about these questions of surveillance, we designed with Chris a set of questions for the panel that will be addressed. What are the gaps, quantitative and qualitative, in the surveillance of emerging viruses? So we said yesterday that sequencing is not the only thing to do to know about viruses. There are other variables that we need to know about behaviors and modes of transmission that are not only quantitative, but that also need qualitative survey. Then how to study transmission back and forth between animals and humans in hotspots of emergence, and how data can be shared between these two realms of health. How to prove that an animal species is a reservoir for an emerging pathogen. How much data do we need on one animal species? Then how to correlate transmissibility and pathogenicity in zoonosis surveillance? And finally, what is the impact of human behavior on viral emergence? And what is the awareness of risk of humans who live in proximity with animals? Now, as a social anthropologist, I'm more concerned by the last question, but I hope that the participants in the panel will address the first questions. And I want to share a research that I've done with a group of anthropologists, precisely on the role of human behavior. So this is a map of all the variables, all the drivers of zoonotic emergence. And you can see that there are a lot of variables, changes in land use, changes in climate and weather. But as we come closer to animal, human, and a vector, we see that interspecies contact plays a major role. And that's what we as social anthropologists can study, how the relations between humans and animals impact the transmission of zoonotic viruses. So I myself studied the perception of avian influenza in South China. And I started from the awareness of risk of emergence when there's a crisis, when there's an outbreak. And when measures are taken to control live poultry markets, such as culling, then consumers realize that behind the food they eat, there is a living being. And I think that in the ordinary work of surveillance, a range of actors are concerned by this tension between two perceptions of the same bird as commodities on the side of consumers, and birds as living beings on the sides of retailers, farmers, food industry, and even bird watchers. And my hypothesis is that microbiologists, veterinarians, and physicians play an intermediary role because they can follow the mutations of the virus between these two status of the same bird. So when we show images of the reservoir for the influenza transmission from wild birds to humans passing through poultry and pigs, we don't have a full picture of all the actors who have different relations to birds. And that's what we as social anthropologists can do, differentiate the modes of relation depending on the position in the food chain. So if you consider birds as living beings, we have farmers and retailers, and the risk is increased by the density of the farms and the markets. And we saw yesterday that closing live poultry market could be a way to reduce risk, but consumers are still attached to this traditional mode of consumption. And then on the side of commodities, we see that people tend to have a contradictory perception. They buy birds, but they also see a spirit of a bird who wants to take revenge against bad treatment. That's something that was used in religions. For example, Taoism, where in Taoist festival, you cut the head of a rooster and you spread the blood on the side of the villages, as on the picture on the left upside. And other religious practitioners release birds to produce merit, with the idea that the spirit of the bird is going to produce good effects on the spirit of the people who release them. And this gave rise to a lot of discussion in Hong Kong and Taiwan and on how to release birds properly between bird watchers and Buddhists. Taiwan has just prohibited the release of birds and also live poultry market. And this tension between the bird as a commodity and the bird as a spirit shows that that consumers need to be questioned on their perceptions of animals, and not only in terms of behaviors. Behaviors can be regulated, but perceptions must be described and influenced so as to produce a good risk management. So what we did with a group of anthropologists was to compare the perception of zoonosis in various societies. And we asked a set of questions to different societies living with animals. How do humans living with animals conceive zoonotic pathogens? We asked if there was a risk of transmission of pathogens. How do they control borders between wild and domestic animals? And how do they manage the bodies of sick animals? Particularly, as we saw, the images of culling have an impact on the risk awareness, but also there is a risk of not being incented to report cases of illness. So the management of culled animals is a major issue. So we had 4 cases in different parts of the world, and I will address them one after the other. So the first case was the Andra virus in Australia, which is the first emerging virus that was discovered on the bat before Nipah and SARS, and after rabies, but rabies had been known for a long time. And it was discovered in 1994 in Andra in Australia, and it caused a lot of deaths among horses and 4 deaths among humans. And so there were massive biosecurity measures to separate bats from horses and humans. And this led to the idea that bats are good reservoirs for pathogens because of their species diversity, because diverse species roost in colonies where they also mixed. Because as mammals, they have adapted their immune system to flight. They have a high longevity because they can repair their telomeres, and they have become more proximous to humans because of anthropogenetic changes such as deforestation or bushmeat. And we were interested, Arnaud Morvan, who did the study, was interested to understand why Aboriginal Australians were excluded from the management of this outbreak, of the Andra outbreak. Because Aboriginal Australians had always lived with bats, as we see with this cave art in Cape York, where the Andra virus broke out. So the question was why there was a possibility that Aboriginal Australians had developed immunity to viruses such as Andra because they had been always exposed to bats. And also, Aboriginal Australians consume bats as a therapeutic product in soups. And they had a lot of techniques to protect themselves from bats when killing them or when using their images, as we see on this painting that an Aboriginal artist made for the anthropologist. And he made some fumigation to protect the painting before killing it. So we kind of asked the question why this group of people who had lived in proximity with bats in Australia for a long time had been excluded from biosecurity measures. And that's a question that we raised for other groups. Now, another group we studied is the herders in Mongolia, which is a post-communist country with 46 million animals and 3 million humans. And herding is the major activity of the country, the third of its economic production. And we studied brucellosis and anthrax and the changing in risk awareness of zoonosis, because brucellosis is a slow endemic zoonosis causing abortions. and that can be mitigated by vaccination and prevention campaigns. And Jacob Zilstag has showed the benefit of associating herders to these vaccination and prevention campaigns, because the measures of killing animals are much more costly if they are not anticipated by these prevention campaigns. So while brucellosis is a slow disease that can be prevented, anthrax is a fast zoonosis, and it is regulated by quarantine and culling. And this has raised debates in Mongolia because of the analogy between anthrax and foot-and-mouth disease. Foot-and-mouth disease is not a zoonosis, but it is controlled by massive killing, particularly in the eastern part of the country. And the massive killing of cattle is considered as inauspicious because the ordinary slaughter of animals requires Mongolian herders not to spill blood on the ground. So all these grounds where the cattle was buried were considered as dangerous. And so the management of foot-and-mouth disease required to avoid this massive killing to respect the popular representations. So what is the perception of animal disease by herders? And how does it involve representations of bodies and souls? So humans have bodies and souls, but animals have only vital breaths. The animal diseases come from the wild, such as foot-and-mouth disease transmitted from gazelles in the eastern part of the country. And the management of this disease requires certain rules to be respected, such as not breaking bones of animals because they contain human souls, and infected carcasses not be buried. Otherwise, it's not auspicious. So that's the result of the research in Mongolia. Now, a third research we've done is on elephant TB in Laos. Doesn't seem to be a major health crisis, but there were warnings after the death of elephants in a US circus in 1996. And in 2011, a survey in Thailand showed that there was a transmission of TB bacteria from elephants to the workers who live with elephants, called mahouts. And this could be related with the prevalence of tuberculosis of 34% among elephants. And Laos was named the country of 1 million elephants, even though there's only about 1,000 elephants today. But elephants are now used in captivity for tourism after the ban of flogging. So the question was there a risk of transmission from elephants to tourists? And when working with mahouts, with elephant workers, our colleague Nicolas Lenné showed that they didn't really, they were not really concerned by the transmission of elephant TB. They had other means of perception of the animal health, looking at their skin and their weight. And so they invoked spirits of the animals rather than the pathogen. And that's why they refused the killing of suspected elephants. Now, fourth cases we studied is MERS-CoV in Qatar. So very famous coronavirus that emerged in the Saudi Peninsula in 2012, infecting 2,500 persons globally with a 35% lethality rate, particularly in the Saudi Peninsula and in South Korea. And camels were found positive to MERS-CoV all over Sahel and Central Asia, but no bats were found with MERS-CoV. So the question was why it emerged in the Saudi Peninsula and not elsewhere. What were the behaviors in this part of the world that may have caused the transmission of this zoonosis? So our colleague Sarah Caballon went to Doha to study the changes in the raising of camels, and she found that camel milk was distributed freely all over the country, that the camel market was bringing camels from all over not only the Saudi Peninsula, but also the world, because some of the camels were coming from Australia, so either for meat or for racing. And camel races are a big hobby in the Saudi Peninsula. There's very little contact because the camels are driven by robots and not by humans, because it was formerly driven by children, but because of the regulation of human rights, children have been replaced by robots. So very little contact, but at the same time, camels play a major role in the life of people in Saudi Peninsula, particularly as they remember them of their past as herders. So an interesting case of recent intensification of relations between humans and camels, because of the growth of the economy in the Saudi Peninsula, that may have caused the transmission of this new coronavirus. So these are different inquiries that we made, and we tried to gather all the information on a framework, and we ask the same question in these different locations: can animals transmit diseases to humans? And as you can see, the answers were quite contrasted, depending on the information people received. So, for example, in Mongolia, a lot of people said yes, and in Laos, most of the people said no. They refused TB to be transmitted by elephants. Whereas in Qatar or Australia, the answers were more balanced. So asking questions about the awareness of the risk is not the best way. We think that we need to do inquiries about how pathogen emerge among many invisible entities perceived by people living with domestic animals, and how these entities enter into tension when there's an outbreak, and measures to control the outbreak, such as culling or quarantine or vaccination. And so we believe that zoonosis revealed the uncertainties of living with wild animals, particularly with this idea of animal reservoir, and that the regulation of zoonosis needs to take into account popular perceptions of species borders. So I'm happy to take questions on this introductory talk, but it also raises questions that will be addressed by the other speakers of the panel.
So thank you, Fred. So now we are open for discussion.
Hi, Frank. Excellent talk. So, I mean, I'm interested you have been talking to many cultural different culture about emerging infectious disease. Very often, there are, I mean, not only about ignorance, but sometimes they actually neglect it or deny there was an outbreak in their herbs or in their population. So they just want to protect their own belongings or their properties. So how can you address this particular problem? Like in terms of disease infectious control, like depopulating whole avian influenza or infected chickens or camels, and that sometimes are very challenging with this type of mentality.
Yes, thank you for this question. So denial can have many reasons. One of the reasons would be the fear to have animals killed without compensation. Another reason may be the attachment to animals, as we can see in camels. Even if camel breeders in Saudi Peninsula are quite rich and could handle the cost of killing the camels, camels are a symbol of their past as herders. So when there is a rapid shift from traditional breeding to more industrial breeding and capitalist economy, then returning to the affects shared with animals is a way to refuse the economy which constrains to killing animals when there's an outbreak. So I guess that compensation would be one issue, one answer, and the other would be to tackle this kind of tension between traditional attachment to animal and considering animals as commodities that can be replaced.
Well, again, I have the microphone. So, Frederick, hi. Nice seeing you, although not in person. I wonder, as Malik also and other people discussed yesterday, what is your impression when you have this type of research on the field of the idea that these measures need to be shared at the level of society so that countermeasures can be accepted? Has any of this been discussed when you give these examples, and what would be a good strategy to have these countermeasures, if not accepted, understood?
My colleague Lyle Fernley has worked in South China on precisely the acceptability of measures for avian influenza in areas where there's been a massive industrialization of poultry breeding. And he shows that particularly around Poyang Lake, which is a hotspot of emergence of avian influenza, and he shows that poultry breeders have developed a double strategy. So they raise poultry at an industrial scale, and they know that it can be destroyed if there is an influenza outbreak. And then they raise small farms with some wild ducks that produce immunity by sharing stocks. They don't use the same term avian influenza for the two scales. So avian influenza is for the industrial scale and not for the local scale. So as Malik said yesterday, poultry breeders don't feel really concerned because they have antibodies, but they are impacted by the countermeasures, and I think they develop strategies to precisely make compatible attachment to small farms and the management of big farms. And then the difficulty is for the bigger audience, the public, to accept these countermeasures because they are often spectacular, such as culling markets, poultry in markets or farms. And I guess that the success of bird release also comes from this the kind of unacceptability of countermeasures, but bird release also raises other risks. So I guess that culling as a regulatory measure should be accompanied by their meaning for the farmers and for the retailers as a way of displaying the strategies that they have. Otherwise, if we focus on the bird, it's on the animals themselves. Then the animals themselves produce a lot of affects of identification and what I call contradictory representation of the animal as a commodity and the animal as a living being.
So Fred, so may I ask one question? So you gave 4 different examples in your talk. So actually, any one of them will be likely, we may possibly become the global outbreak. So in this kind of case, different countries, they have different understanding on a particular of animal. So is it, from your point of view, is that means it is very hard to have a common agreement or common approach of a countermeasure whenever an outbreaks occur?
Okay, so we chose these 4 cases because our colleagues had the knowledge about ordinary relations with animals in these 4 countries. Even for Muscovy, we work with an anthropologist who had studied camel breeding in Central Africa, in Saharan Africa. before. So we, as anthropologists, we cannot come after the outbreak because then we arrive at the moment of emotion and criticism and accusations. We need to understand before the outbreak how people live with animals, so that we can really say how these countermeasures, such as vaccination or culling, can become acceptable. And then I must say that surveillance is also one of the countermeasures. I mean, the best way to tackle zoonosis is to avoid them, to anticipate them, and mitigate their effects. And so that's why hiring, enrolling farmers in the surveillance is really one of the major issues, precisely to avoid this affective reactions to strong countermeasures.
Any question from online? Yeah. Okay, Mary. Yeah, go ahead.
Yep, we have a couple of questions from online. First one is from Zhongli Shi, and she's asking, is it possible to develop some models to predict the risk of potential zoonosis?
So I've not heard the question online, but because I'm supposed to read the questions online for the panel, I'm able to read them on WhatsApp. Have you measured on? Yeah. So is there an intensification is there a change in relation with animal because of increase in biosecurity measures, if that's the question? I guess that Mongolia would be an interesting case because the country is more and more exposed to international trade after the fall of the Soviet Union. And at the same time, it raises pastoral techniques as a kind of heritage that needs to be defended because of the role of herding in its economy. So there has been an increase in biosecurity measures, but at the same time, there has been a kind of valorization of herding techniques. So the people are more aware of the risk, but they also want to defend their traditional mode of killing, such as not spilling the blood on the soil. So it's not only an adaptation to biosecurity; it's also a way to find mitigations that makes this measure acceptable.
So is there any question? So first of all, Marie, is there another question, right? Yeah. Yeah. So Vijay can ask.
Thank you. A great talk, Fred. I mean, the focus, much of the anthropology studies focus has been on Asia and Africa, where there's been a historical development, long-term agriculture, long-term practices. I was wondering if there were anthropological studies towards Western countries where there's a shift from industrialization towards organic farming, backyard poultry, and the increased risk of exposure to these animals, to wild birds, for example, and things like that. So are there other studies studying these newer developments in the West where there's an increased risk of exposure towards avian influenza, for example? Did you hear that?
So the question is if the shift to organic farming has raised some new risk of avian influenza?
That's right.
Okay.
I mean, so, yeah.
Okay. Well, for example, in Hong Kong, there are 30 poultry farms. And despite the voluntary surrender scheme implemented by the Hong Kong government, the consumers still buy poultry as a kind of organic food, which is more expensive than in China. So Hong Kong would be the good answer to your question. Other anthropologists who have worked in Thailand or Vietnam have showed that biosecurity measures, by reverse, encourage the development of industrial farms because small farms do not have the capacity to implement biosecurity measures, which are quite costly in their farms. So Hong Kong is quite an exception at this level, and we currently have outbreaks of H5N8 in the south of France, in the area where we produce foie gras. And it's also the bigger farms that are concerned because they have concentrated their poultry stock, whereas the smaller farms where poultry is raised in open air are affected by the H5N8. The bigger farms with their big concentration are the places where it actually is amplified. So we currently have a big debate whether we should keep high concentration or if we can also keep maintain some open air breeding.
Hi, Frederick. Yes, we had a second online question as well about from Zhongli Shi asking, is it possible to develop some models to predict the risk of potential zoonosis?
So it would be interesting to develop models comparing precisely the impact of small scale and big scale, because scaling has had an impact on the emergence of zoonosis. As I mentioned, in China, small scale produces risk, but also allows farmers to mitigate the risk, whereas big scale leads to massive culling. So I think we should correlate the scale with the risk of emergence and with the acceptability of countermeasures. That would be a good model to build.
Maybe we should move on to the next sections. So thank you, Fred. And I would like to emphasize that Fred will be also the moderator in the panel discussion in this round. So in this session, so we are coming with the panel discussion. So we have invited 3 different speakers, and every one of them will give a short talk and then come up with the Q&A section. The first speaker that we invite is Dr. Véronique Chevalier, who is an epidemiologist and a vet with work experience in Europe and Africa. Her research mainly focuses on emergent disease, epidemiology, and modeling, especially on the vector-borne diseases. For example, the Japanese encephalitis or Rift Valley fever. She's currently a principal investigator in the epidemiology and public health unit of the HKU-Pasteur Cambodia. Her topic today is integrate surveillance of potential zoonotic beta coronaviruses in the wild animal value chain in Cambodia. The second speaker is Annabella Yu, is an entomologist with many years of experience on mosquito. Her research focuses on understanding the interaction between different arbovirus, such as chikungunya, dengue, RVF, West Nile, yellow fever, and their mosquito vectors. She is currently the head of the arboviruses and insect vectors unit at the HKU-Pasteur, Paris. The topic that Annabella will going to share with us is the threat of yellow fever in Asia, the entomologist's point of view. The last speaker is Dr. Crystal Liderius, who is a medical anthropologist in the University of St. Andrews. His research focuses on the anthropological and historical examination of multiple aspects, such as epidemics, zoonosis, pandemics, as events posing as existential risk to humanity. His topic today will be the ethnographic for pandemic response, post-bats, challenges and limitations. So I would like to first invite Veronique to have her talk.
Well, good afternoon, good morning, or good evening, everybody. Can you hear me?
Yes.
Yes, it's okay. So, first of all, I would like to thank Hong Kong University and Hong Kong Research Pole for the organization of this workshop and for giving me this opportunity to give this talk. So I will during this talk, I will present briefly a new project which is called ZooCov, which aim at developing an integrated surveillance of zoonotic beta coronaviruses in a wild animal value chain in Cambodia. So as an introduction, I just would like to remind that emerging diseases surveillance is challenging because it's challenging to know how to monitor something that sometimes we don't know. So we have several options. We can monitor the pathogens in wildlife. We can monitor the pathogen in vectors, or we can monitor the abundance of vectors. We can work on domestic animals. We can work on humans. Sometimes we even work on unknown pathogen or on disease for which the epidemiology is unknown or partially known. So the modeling, prediction, and risk-based surveillance are not really applicable. So it is challenging. So we have still traditional surveillance tool. So the first one is the passive surveillance, which is based on the reporting of so-called cases. The problem is that when we work with wildlife and pathogens that are asymptomatic in wildlife, this is not applicable. We have the systematic or the active surveillance, which rely on regular sampling. This is very efficient. That can be very sensitive, but it's also very expensive and time-consuming. We have the syndromic surveillance, which rely on the report of health-related event. That can be fever, for instance, or that can be care-seeking behavior. Again, it's very sensitive, but it's not specific at all. And we have the participatory surveillance, which involve citizens, and which is really useful to be able to benefit from the knowledge and the experience of local communities. So the perfect surveillance system is probably a combination of different methods depending on the knowledge we have about the disease we are monitoring and the objectives. We've always a trade-off between the perfect sampling and the logistical, ethical, legal, and financial constraints. So ZooCov is a new project that we started to implement in Cambodia, and which aim at developing an integrated surveillance of beta coronaviruses in the wild animal value chain, in Cambodia. So why Cambodia? Dr. Vesna Duong mentioned it yesterday already. So in Cambodia, there are some viruses with pandemic potential, and the SARS-CoV-2 ancestors have been isolated recently from 2 bats that were sampled in Cambodia in 2010. In Cambodia, there are also the primary reservoir. There are large colonies of rhinoloph bats. We have also the potential intermediate reservoirs. We have civets, pangolins, rodents, and maybe other intermediate reservoir that we still don't know. And in Cambodia, there is also a wildlife-human interface because there is wildlife trade and bushmeat consumption. So all the components of emergence are in Cambodia. So ZooCov is really a pilot survey, a pilot project, which is implemented in Cambodia in 2 provinces, in the Mondulkiri and Stung Treng, which are two provinces that are where there are still big forests, protected areas, and wildlife. And it's implemented with the support of the Ministry of Health, of the Ministry of Agriculture, and the Ministry of Environment of Cambodia. And the project relies on 4 pillars that are interconnected, of course. So the first pillar is an existing wildlife health surveillance system, which is piloted by the Wildlife Conservation Society WCS in Cambodia. And this surveillance system is based on the reporting of mortality and morbidity in wildlife. The second pillar is an analysis of the main wildlife value chain in Cambodia. The third one is a deep analysis of the practices and the perceptions of people who are involved in this trade about the risk of handling, hunting, or consuming wildlife. And the last pillar is a documentation of the beta coronavirus's diversity, which are hosted by the animals that are either sold or hunted or consumed in Cambodia, and the quantification, of course, of human exposure to these viruses and the determinants of this exposure. For the first component, we use the participatory epidemiology, focus group discussions, and immersion. And this was really a step-by-step process, going from informal discussions with people that are involved in the trade, to move to more formal discussions, to more formal focus group discussions or face-to-face interview. To establish first a list of actors, of people who are involved in this wildlife trade, and then to move forward an analysis of the relationships between these people, the relationships that can be commercial, that can be political, or that can come also from family or friends influence, or from the regulation influence. So this first component took a lot of time to get contact with people and to get the trust of people. The second component, so we use this stakeholder map to identify a set of exposed and non-exposed people to wildlife. We have in this batch more than 600 people. So we've been working with rangers, consumers, butchers, hunters, forest workers, and others. And we interviewed all these people face to face. And we used for that dedicated questionnaires that were built in collaboration with socio-anthropologists to try to get more information about their motivations and their barriers about wildlife consuming or hunting or trading. That can be the price, that can be the social position, that can be the taste, that can be the use for traditional medicine. On the other side, that can be they cannot consume wildlife because of the fear of disease or because of the fear of the police. And we used also this questionnaire to establish a typology of the contact with wildlife in terms of species, in terms of frequency, and in terms of type. Is it during handling? Is it during butchering? Is it during consuming? And then all these people that we interviewed were sampled twice at a 6-month interval. So we are in the process of the second sampling session now. So for all these people that are sampled, we have a lot of information. We know their age, their sex, we know their position, we know, of course, the perception we have about the risk of handling the wildlife. We recorded also their traveling history, of course, and we also recorded any clinical signs that may have occurred during this 6-month interval. And at the same location and at the same time, we implemented a long-term opportunistic sampling of confiscated or dead wild animals that are, these animals are confiscated by NGOs that are working in this location. And at the same time, we implemented targeted sampling of rodents and bats. So all these samples are going to be analyzed by the Pasteur Institute of Cambodia by serology. So we have a test which is able to identify antibodies against the more pathogenic beta coronaviruses. This test is adapted for humans, domestic animals, and and wildlife, including bats. And the swabs coming from the wildlife will be also analyzed by PCR and sequencing in IPC. The last step will be statistical modeling to identify drivers or predictors of human exposure to these viruses, and maybe also to identify some determinants of the beta coronaviruses detection, maybe species, maybe location, or maybe the season. We'll see the result we will have. And the next step and the ultimate goal of the survey is to move from let's say the traditional wildlife surveillance piloted by WCS into a more integrated surveillance system of the beta coronaviruses circulation or spillover from wildlife to human. So the first step, and we are in the process of that, we are in the process of evaluating the existing wildlife health surveillance system. So we work with some criteria for which we have some data to qualitatively analyze this data. So we work on the sensitivity, of course, of the system, but also we work with the beneficiaries. We are looking at the constraints, at the influence, at the resources, at the adaptability of the system, the leadership, and the stability. The next step will be to use participatory modeling sessions to refine the system, to try to improve it, and of course we do it with in close collaboration with Wildlife Conservation Society. So we will work first on the objective, the data collection, and the methods. We will see whether it would be relevant, for instance, to incorporate syndromic surveillance on exposed people, to incorporate wildlife feces collections that can be more easily done than traditional sampling of wildlife or other methods. We will work also on the components and the structure, meaning that we will work with the other actors to see how to integrate in this surveillance system the human health sector and the environmental sector also, and probably more important, the local communities who know perfectly their ecosystem. They know perfectly how the things are working, and they are probably the most efficient people to monitor something in this ecosystem. And we will work also on concerted modalities of collaboration between these sectors, so human health, animal health, and environment, and the actors. And the next step, because this wildlife trade is illegal in Cambodia, but most of the time tolerated because it's coming from this trade has sometimes very deep cultural roots, and also this trade represents a major income for people and a source of protein. So we have to also keep in mind that this system to be efficient need to be acceptable for people and need to be sustainable for the people and also for the country. Thank you for attention, and I will be happy to answer your question.
Again, Veronique, hi, this is Roberto. I have the microphone. So while people are thinking about the next question, I will ask the first. Can you hear me? So why, I mean, if did you start and did you decide to focus on coronaviruses after or before the emergence of the SARS coronavirus 2? Just to try to find out how was your choice motivated?
Okay, thank you for your question. The sound is very bad, but I think that you asked me why we choose coronaviruses. Was that the question?
Yes, and whether you yes. selected this before or after the beginning of the pandemic.
Okay, it was so this project is granted by the French, the National French Agency for Research, that released the call for project for fundings. It was in February 2019. It was just at the starting of the pandemic. And I remember that we submitted the proposal in March, and this is why, of course, we choose the coronaviruses on one side, but on the other side, also because here in Cambodia there is a long history, let's say at least 10 years history of research on coronaviruses, on bats, Nipah virus, where Dr. Vesna Duong presented that yesterday. So we have a long-term history of collaboration with Wildlife Conservation Society, with Pasteur Institute, of course, and this is why we choose the beta coronaviruses.
So Veronique, so how will you transform your data to become the government policy on controlling the virus?
I don't hear anything. Maybe Frédéric, do you hear better?
So can you hear? So you can hear?
It was okay.
Yeah.
No.
No. No.
I don't hear anything.
Can you hear from this microphone better?
It's a little bit better.
Okay, maybe you can try this, Chris. We need another microphone.
So Veronique, so you can hear, right? So how do you transform your integrated data into something that the government can use as a policy to control a particular virus?
That's a good question. I don't think I have the answer. The goal of this project is really on one side to improve the knowledge we have about the diversity of beta coronaviruses that are circulating in Cambodia. This is one side of the project, and the other side is to provide new knowledge coming from let's say the biological components of the problem, and some more knowledge also about the way people are behaving, and let's say to try to quantify the risk, and then see how we can integrate that in the surveillance system. So we are only at the beginning of the project. We are still collecting the data. So what I hope is that we will be able to quantify the risk. We will be able to identify the people who are at risk, hopefully to implement some surveillance strategies, maybe through syndromic surveillance in hunters, for instance, or it will really depend on the result we will have.
We have a question online from Peter Rotier in the Netherlands. Do you see opportunities for wastewater surveillance, as we have in the Netherlands, from cities to monitor SARS-CoV-2?
In animals or in?
I think it would be in cities.
In cities.
So.
Why not? I would say because surveillance can be. We can imagine having some kind of syndromic surveillance in humans. Yeah. In cities, that can be a very efficient tool. The problem of syndromic surveillance is that it's very sensitive. If you are monitoring, for instance, fever syndromes, you will have a lot of positive cases, let's say, but probably a very small fraction of these syndromes that will really correspond to what you are looking for. So it's very sensitive, but not specific at all. So that means that you have in the background, you have to be able, from a lab point of view, to receive all the samples you will have and to treat all the samples you will have.
So Veronique, so Fred, you have a question from online?
Yeah, I don't have other online questions, but I wanted to add a question to Roberto's on the impact of the COVID-19 crisis on the awareness of risk for those who are in contact with wildlife. Can you assess that? from previous studies that have been made in Cambodia.
Sorry, that was a question for me. I thought it was a question for Roberto.
No. It's following on Roberto's
Roberto.
question. No, it follows on Roberto's question, but it's for you.
Sorry. Can you repeat, because I thought it was for Roberto. Sorry.
How can you assess the impact of the COVID-19 crisis on the risk awareness of people in Cambodia living with wildlife?
Yes, we have in the questionnaire a part which is dedicated specifically to this assessment. And as far as I know, because we are really in the process of starting the analysis of the questionnaire, some people are really aware of the link, of the potential link between COVID and bats, let's say, and some people clearly told us that they stopped hunting bats or hunting civets or hunting animals, and some people are not aware at all. So it really depends on the people we are working with. We interviewed let's say 10 different classes of people. So we have official, we have authorities, we have hunters, we have people that are living in the forest. So obviously these people are not aware of even not aware of the COVID crisis. So it really depends on the people we are working with.
So I have another question. So I expect you will find lots of virus from those wild animals through sequencing. And then how do you use this information for your serology test? Because you expect some of the virus may not be easily cultured, or you just have the sequence. So how do you understand the interaction of this virus to human through the serology?
Sorry, I don't hear anything. Can you just be? I'm sorry, the sound is awful.
So here, can you hear?
Yeah.
Yeah. Okay.
Yeah.
So I expect that you will find lots of different viruses from your sequencing in the wild animal. So how do you perform the serology test from those findings? Because you may expect some of them, they may not be cultured. And then what will be your approach?
So for the serology, we used an assay which was adapted from a work that was done by Martin Peters. And we here in IPC, I'm not a virologist, so maybe I will not I'm probably not the most competent person to answer you, but for the serology, we used a test which is able to detect, if I remember well, 6 or 7 types of antibodies against coronaviruses. And this test has been adapted to human, to domestic animals, and to wildlife, including the bats. This is for the serology, and for the wild animals that are confiscated, the bats that we are sampling and the rodents. We take rectal swabs. We take blood when we can. So the blood will be analyzed using the same serological test, and the swabs are analyzed with PCR here in IPC and sequenced.
I mean, any other question here? Fred, any question from online? If no, thank you, Varek. Yeah. So the next speaker will be Annabella. So she will share her view as an entomologist. So please.
So hello everyone, and I'm Annabella Fayou from Pasteur Institute in Paris. So I am entomologist working in the Department of Virology. So I will talk today on emergence and arboviral diseases, and mainly yellow fever in Asia. So very quickly, some keywords to better understand what is the risk in terms of a transmission of arboviruses. So there are around 500-600 arboviruses, and half of them are transmitted by mosquitoes, and 100 are passed then to humans. So the most important arboviruses for the human health are dengue, Zika, chikungunya, and yellow fever. And you can see in this map, they have been all isolated for the first time within a zootic cycle. And several decades later, 70 years later, they all spread outside their natural regional distribution and colonizing most tropical and even temperate regions, including Europe. So most arboviruses circulate primarily within a zootic cycle between non-human primates as reservoir hosts and arboreal canopy-dwelling mosquitoes as vectors in a forested environment. And then the emergence coincides with the capture of a white virus by an anthropozoophytic mosquito, which acts as a bridge vector for transmission of the virus from animals to humans. And then the virus circulates within an epidemic cycle, with humans as both reservoir amplification host and anthropophilic mosquitoes, Aedes aegypti and Sabopictus, as the main vectors. So the two mosquito, Aedes aegypti and Aedes albopictus, are vectors on the 4 arboviruses that affect the human health, dengue, chikungunya, Zika, and yellow fever. And the 2 mosquito species can also share the same geographical distribution. In this map, you can see countries when we have only Aedes aegypti here in blue, region where we have only Aedes albopictus in orange, and countries where they are both together here in beige. So all mosquitoes are not able to transmit all arboviruses. What makes a mosquito competent to transmit a human pathogen? There are several criteria, and mainly 4. The first one is that the mosquito should be in high densities, abundant enough in the human environment. The second criteria is that mosquitoes should bite humans, so be anthropophilic. And the third criteria is that after the infection, the mosquito should survive long enough to be able to transmit the virus by bite. So the 3 criteria, 1, 2, and 3, can access through entomology survey on the field. And the 4th criteria is what we are looking at in the lab, is the vector competence. So what is the vector competence? So vector competence is the ability of an insect to ingest the virus during a blood meal, to ensure the viral replication, and transmit the virus to a suitable vertebrate host. So the duration between the entry of the virus inside the mosquito, the exit of the virus from the mosquito, is named the extrinsic incubation period, the EIP here. So it's the time necessary for the virus to reach the salivary glands and to be transmitted. So concerning the yellow fever is an old disease. It's a disease endemic to tropical regions of Africa and South America today. And both yellow fever and Aedes aegypti are both originated from Africa. So during the slave trade, they have been introduced into the New World. So you can see on the left, a phylogenetic tree showing that yellow fever from South America here are closely related, genetically related to yellow fever from West Africa. This is one clue. On the right, you have a migratory route of Aedes aegypti from outside Africa into the New World, and this study has been based on population genetics analysis. So despite the availability of an efficient vaccine, the 17D, several outbreaks have been reported recently, and mainly in Africa and South America. And you can see, for example, in America, it was in Brazil in 2016, and in Africa, in Angola in 2016. So in collaboration with other colleagues from Central Africa, in Cameroon and in Gabon, from the Center of Research in Infectious Diseases, we have evaluated the vector competence of different populations of Aedes aegypti collected in different regions in Cameroon and Gabon, and estimated the vector competence when infected with the virus from Africa. And you can see here that for Aegypti populations, that all the mosquitoes are able to be infected, to disseminate where the virus, and to transmit the virus with the virus detected in the mosquito saliva. This is for Aedes aegypti. What about Aedes albopictus, which is a new mosquito in Africa? So you can see that this mosquito is less competent to transmit the yellow fever virus. So it seems that Aedes aegypti behave as an efficient vector of yellow fever compared to Aedes albopictus. So in Brazil, after the eradication of the mosquito Aedes aegypti, yellow fever disappeared from most cities in Brazil and only persists a selvatic cycle of yellow fever here in green. And this region is approaching slowly the densely populated cities in the southeast of Brazil, including the city of Rio, where the vaccination against yellow fever is not mandatory. So then the question is, can mosquitoes contribute to a kind of reurbanization of yellow fever like in the past? So, including Aedes albopictus. You know that Aedes albopictus can feed on humans, can feed on animals, so can play the role of a bridge vector for transmission of the virus from animals to humans. So to answer to the question, we did passages of the virus, yellow fever virus, on two populations of Aedes albopictus. Aedes albopictus from Manaus, from a region endemic for yellow fever. We also use Aedes albopictus from Rio, a region free of yellow fever. So we infect the mosquitoes. 21 days after, we collect the saliva from individual mosquitoes, pull all the saliva together in the same pool, and then grow them on cells, obtain a high titer of virus, and then give it back again to the mosquitoes. So we did 10 passages, and after the 10 passages, we give each passage to mosquito to evaluate if the passage can be transmitted by the mosquito which has been infected. So you can see here in this graph that from the passage 1 to the passage 4, there is no virus detected in the saliva of the mosquito. But from the passage 5, you can see that the virus can be excreted from the mosquito which has ingested a virus that has passed several times on Aedes albopictus from Rio. And you can see also here that the virus can be transmitted when we infect the mosquitoes from Manaus. So we can say, using our experimental procedure, we were able to select a yellow fever virus well adapted to Aedes albopictus, which is an unusual vector of yellow fever. So to answer the question, can this mosquito contribute to the reurbanization of yellow fever? The answer can be yes, because this mosquito is present close to the city and also at the fringe of the forest where the wild cycle of yellow fever is maintained. So during the last decades, several outbreaks have been reported of yellow fever. For example, in Angola, with an outbreak with 3,000 people infected. And important cases were reported outside Africa, including in Asia and in China, and where the vaccination against yellow fever is not mandatory. So what we did, in collaboration with our colleagues of the Asian Pacific region, we have evaluated the vector competence of 12 populations of Aedes aegypti from Asia and Pacific region, and we evaluate the vector competence after the infection with yellow fever from Africa. We evaluate at the 14 and 21 days after the infection. When you examine the infection of the midgut, which is the first barrier to the entry of the virus inside the mosquito, you can see that all populations were able to be well infected. When you examine the dissemination of the virus beyond the midgut barrier, so with an efficient crossing of the midgut, you can see that most populations also were able to disseminate well the virus. And when we examine the transmission by detecting the virus in mosquito saliva, you can see that all the populations were able to transmit the yellow fever virus 21 days after the infection. So in this case, the 12 populations of Aedes aegypti from Asia were able to transmit yellow fever from Africa. So concerning Aedes albopictus, which is an Asian mosquito, We evaluate also the vector competence at day 14 and 21 days after the infection. And you can see that the infection of the midgut is less efficient compared to Aedes aegypti. When you examine the dissemination, also in this case is less efficient. And when you examine the transmission by detecting the virus in mosquito saliva, you can see that only one population at the 21 post-infection is able to transmit the yellow fever virus. So we also examine the viral load in the single saliva of mosquitoes from each population studied. And you can see that in some cases, we can have a viral load reaching more than 10 to the 4 viral particles in the saliva. And when after that you compare the viral load of the virus detected in the saliva of mosquitoes Aedes aegypti from Asia, compared to mosquitoes from Africa and compared to mosquitoes from Pacific region, you can see that mosquitoes from Asia presented viral loads comparable with the viral loads estimated from mosquitoes from Africa. So we can say that Aedes aegypti from Africa are as susceptible as Aedes aegypti from Africa to transmit yellow fever virus. And you also did some modeling using to evaluate the transmission risk through Aedes aegypti, using the vector competence data and also the occurrence of the mosquito in different regions in the Asian Pacific region. And you can see here, for example, in the CSP, it is here on the left hand in the map. In Thailand, you have a higher risk of transmission of yellow fever through Aedes aegypti. So to conclude from my talk, we can say that the emergence of arboviruses appears as a process in 2 main steps. The first step is the introduction, adaptation of the new vector to a new biotype. And the second step is the introduction, adaptation of the new virus to a new vector. In the case of Asia and for the yellow fever, we have Aedes aegypti already susceptible to infection with yellow fever. You have the people which can be sick, those are susceptible to the yellow fever infection. The virus can be introduced, and the environment is permissive for maintaining a transmission cycle. So it seems that in Asia, all the ingredients are present to trigger an emergence of yellow fever. So to finish, I would like to thank all the members of my team and also my collaborators for this work, and also the funders who has contributed to this work. And I'm happy now to take your questions.
Thank you, Annabella. So open for discussion.
It's an anonymous question. Could there be a risk of antiviral resistance or adaptation that enhances pathogenesis arising from re-urbanization of yellow fever?
So Frederic, it's a question for mosquito or for humans, antiviral in humans. Antiviral humans, okay. So we did this work in the previous time using antiviral against chikungunya, and we give these antivirals to mosquitoes and to see what happens. And because the antiviral also can select the viral population that can resist the antiviral. So the question is, can these new strains selected by the antivirus be transmitted where by the mosquito? So we gave to the mosquito these new strains, and we found that hopefully that this resistance trait was not transmitted by the mosquito, because there is also a risk to use overuse of antivirals can generate resistant strains, and then can be disseminated by mosquitoes. That can make the situation worse, and so this is one point very important.
And a second anonymous question: why do we see transmission and mosquitoes that can transport viruses in countries like Mauritania without seeing the same in neighboring countries like Western Sahara, although they have similar weather conditions?
they have a similar weather or conditions, and the environment is not the same, and also the virus strain is not the same. So there is a multiple factor that can explain the profile, which can be different between the two neighboring countries. But one point that should be underlined is the mosquito. The mosquito is, they have different populations who are genetically different with different susceptibility to the infection. And also the mosquito breed in some sites. The breeding sites are, the composition of the breeding sites can be different in terms of microbial loads or if there is some presence of insecticide particles. We have some selective pressure that can perhaps change the composition of the mosquito population. And in this aspect, the microbial composition of the mosquito is very important, because it can also attenuate or mitigate the transmission, in the sense that we have, for example, the example of Wolbachia, which is a bacteria widely used to control dengue. And these bacteria interfere with the replication of some arbovirus, including dengue, yellow fever, and others, and making the mosquito less prone to deliver the virus when the mosquito is biting. So that's an example saying that the bacteria inside the midgut, which is acquired during the immature stages as larvae in the water, can also interfere with the transmission of arboviruses for the adult stage.
Well, yes, if there is now time, Annabella, hi, this is Roberto. Can you hear me?
Yeah, Roberto,
okay.
I can hear you.
All right. So, having established that there is vector competence for yellow fever, from your point of view, what bad situation can arise to have an outbreak similar to the one in Angola, with several thousand cases and several hundreds of deaths? So what scenario could lead to this type of event? Because yellow fever is a very serious disease, but luckily, it gives symptoms, so it cannot pass, without unnoticed.
So Roberto, we are talking about the situation in Hong Kong.
Or no, in Asia in general.
In Asia in general.
Because you talk about vector competence in Asia.
So you can see that from the experiments that Egypti is more susceptible to the infection than Aedes albopictus. So in a region where we have only Aedes albopictus, the risk of getting an outbreak is minimal. But you can see that, so for example, in Hong Kong, we have only Aedes albopictus in Hong Kong, but Hong Kong is surrounded by countries where you have Aedes aegypti, and is an invasive species, and they got to compete at its advantage on Aedes albopictus. So one day Aedes aegypti will come in Hong Kong, but you can see outside you have aegypti. But I think that if we increase the trade with Africa, we increase the chance for getting more and more people infected with yellow fever. And one recommendation is really to vaccinate all the people going to Africa and coming back, to make sure that we have no risk of getting back some virus. And this is the main advice to give, because the vaccine is, the 17Ds is super efficient. And why not using it more widely in Asia for people going for work or for others, for tourism in Africa and South America.
Our next speaker is Dr. Christos Lentiris, who is a medical and pathologist. So please.
Hi, good morning from the UK, and thank you very much for this invitation to talk at this workshop. And my talk is going to focus a little bit more on what Frederick said earlier today about the role of anthropology in studying epidemics and zoonotic diseases. And we may call this, that's not actually a term used in anthropology, but it's kind of a nice short way of putting it. Let's call this epidemic ethnography. Now, epidemic ethnography has two, if you want, main aims. One has to do with interpretation, and the other has to do more with applied context of response. And in terms of interpretation, of actually interpreting what's happening on the ground, a key aim of anthropology, be it social anthropology, cultural, or medical, is to go beyond the medicalization of culture. We have seen medicalization of culture in the context of several outbreaks, SARS, Ebola, and now COVID-19. And by this we mean processes through which different cultures, usually non-Western cultures, are seen as inherently pathogenic. Bushmeat markets, wet markets, so-called wet markets, etc., are being medicalized in Orientalist and rather colonialist ways, which completely fail to understand processes on the ground and create very difficult situations when it comes to epidemic control. At the same time, however, medical anthropology aims to go beyond the culturalization of epidemics. And by this we mean the tendency, which also exists in epidemiology and epidemic response, to attribute patterns, say, of compliance or resistance to epidemic control measures to culture. And again, culture here is usually non-Western culture, which again is a very racialized way of approaching human behavior and social responses. When it comes to actually proposing something, medical anthropology and anthropology in general is interested in fostering not just community responsive approaches to epidemic control, but community-led approaches. And the Ebola outbreak of 2014-2016 in West Africa has been one of the main grounds where these approaches have been developed. And there are several excellent studies of this sort of approach. Now, while these two legs of anthropological approaches to epidemics have to do with epidemic emergencies, anthropology is also interested, as Frederick mentioned, in more long-term processes, not so much what happens during an epidemic or a pandemic, but the long processes which may lead to infection or to actual outbreaks. And anthropologists are very interested in understanding these long-term processes of emergence and infection, precisely when it comes to zoonotic diseases, at this interface of what Anne Kelly and Hannah Brown have called material proximities between humans and non-humans, but also on the symbolic and ontological levels of this interaction. In practical terms, anthropologists are also interested in fostering sustainable, resilient, and community-led frameworks of prevention and preparedness. So this is not just about these approaches when an outbreak happens, but more to create long-term sustainable patterns in different societies that would allow for the prevention and preparedness for infection. Sorry, I'm trying to go down. So what is really important in both these processes is what medical anthropologist Charles Briggs from Berkeley has called communicative justice. And this means anthropologists' ability to facilitate and foster voices from the ground, which are usually not heard. Voices which come from marginalized groups or people in a given community which are not given a voice within institutional settings, and which may have both vital information for us when we're examining disease emergence and zoonotic infection, but which also need to be heard in order to guarantee equity on the ground when it comes to epidemic control. At the same time, in recent years, anthropology has been interested and very active in developing interspecies approaches to epidemic control. which are not destructive to the social fabric of affected communities. This means anthropology is interested, as Frederic said, to understand how people on the ground in different societies approach animals, relate to animals, what is an animal to them and what is not an animal. And this is of interest to anthropology in and of itself, of course, because we are an ethnographic discipline, but it is also very important for epidemic control, for epidemic prevention, because only if we understand how people on the ground relate to these animals, are the animals just animals, or are they ancestors, are they spirits, what sort of spirits, what different animals do what, only then can we understand how zoonotic and emergence patterns emerge in specific settings. Now, when it comes to an actual outbreak, taking responses on the ground ethnographically seriously is of key importance to epidemic control. And this means to take responses to a given epidemic and to epidemic control beyond the usual behavioral dichotomy of compliance and resistance, which unfortunately we have seen plaguing understandings of how different communities respond to COVID-19 in the past year. This means to take seriously the complexity of attitudes, the cosmological patterns, the sociological and historical drivers of these responses, rather than trying to box everything into this black and white plus minus compliance resistance pattern, which is kind of epistemologically speaking, completely invalid in all societies, probably, but definitely in societies which do not actually adopt such dichotomies, such as societies outside the Western world. And the hope in this is that this will help us reimagine global health as something which is inclusive of different perspectives and not simply Western ones, to decolonize global health, and to create nuanced understandings of zoonosis as a process which involves not simply a mechanistic transference of a pathogen from a non-human animal to a human being or a human community, but incredibly complex patterns of animal-human relations on the ground. Now, these objectives pose some very interesting challenges. Meeting these objectives requires us to understand how is ethnography produced in response to epidemic emergencies. And ethnography, as you may know, is usually long-term. It takes at least 12 months. So when we produce ethnography in response to epidemic emergencies, we have a rather unusual situation when ethnographers or anthropologists are called on the ground to produce knowledge or data about an outbreak and how societies are involved in it almost immediately in very short-term boundaries. And this is methodologically very unusual for anthropology. So we need to consider what are the distortions involved in such rapid production of knowledge or data. But we also need to consider how already existing ethnographic data get transformed and translated into epidemiological evidence in the context of these emergencies. Data that have been collected not for the purpose of understanding epidemics, but say kinship systems or rituals, are often translated into epidemiologically meaningful evidence in the context of emergencies. And this is a very difficult process, not an automatic process in any way, and one which we need to discuss very seriously so as to make sure that the integrity of the originally collected data is not lost in the name of the emergency and is not distorted as a result. With Frederic and other colleagues, we have been very interested in the past years to examine this question of what are the epistemological, ethical, and political implications of translation from ethnography to epidemiology. And key to this complex network of questions and challenges is a kind of very old-fashioned anthropological question of who speaks for whom. Anthropologists usually talk with people, and then they speak about them, and sometimes in situations of advocacy for them. And this sort of translation of ethnographic data into epidemiological evidence involves epistemological problems, such as what counts as evidence for anthropology and what counts as evidence for epidemiology, but also ethical questions. Because in many ways, when we have gone down to the field to collect data, this was not meant for public health purposes. And we do not have actual ethical clearance to use our data for these purposes, but also political implications, such as who benefits from this data. Is it the actual communities which are being, which were studied, or is it someone else? And how are these communities disadvantaged by this use of the data? On a more epistemological level, a key challenge is how are different kinds of evidence integrated in understanding and controlling, but also communicating epidemics and epidemic risk. As I said before, it may surprise you to hear that anthropologists do not consider evidence to be an empty category. It is a category which is always already entangled with ideologies, with politics, with cosmologies and ontologies. And in this way, we are very self-reflective about categories of data and evidence. And when it comes to integrating different kinds of evidence, evidence produced to different purposes and within different epistemological frameworks, anthropology is always very, very careful, because the tendency that we usually see in such integrations is a rather aggressive positivism with which anthropologists would generally feel very uncomfortable. Now, these sort of questions, in order to be addressed, require new types of collaboration on the ground between life scientists, virologists, epidemiologists, and anthropologists. It is my opinion that this collaboration needs to be non-teleological. Any meaningful collaboration between disciplines needs not to be driven by an already decided answer of sorts, but needs to be open-ended. It needs to be non-extractive in the sense that we need to be respectful of the communities which we are studying and always be in dialogue with them in the ways in which we use the data extracted or collected from them. And it is a collaboration that needs to focus not exclusively on finding answers, but on much more on forging common questions and on unsettling existing questions and unsettling epistemological frameworks of both anthropology and epidemiology when it comes to infectious diseases and zoonosis. This is, of course, a huge challenge, and I hear you telling me this is not the time for this, this is an emergency. But I think that actually to adopt an ancient Greek terminology, an emergency is actually an opportune time. It is the kairos for achieving such unsettlements. Because as we saw with Ebola, I'm afraid to say, the collaborations and the wonderful interdisciplinary unsettlements and meetings that happened during the Ebola outbreak withered away once Ebola disappeared. And this is always the challenge and the problem with epidemics, that epidemics last in terms of collaborative frameworks, mostly during emergencies. And then everyone goes back to their own discipline in order to calculate impact and all these things we need to do in academia. So I would say the emergency of COVID-19 is precisely when we have the opportunity to pose these very important questions on collaboration and on what can ethnography do for epidemiology and epidemic control. Of course, this is a very pragmatic and important question, but also how can epidemiology and anthropology come together in redefining global health and redefining zoonotic disease and infection. Thank you very much.
Thank you, Christos. So questions?
Well, Christos, I'm happy to start again. Thank you for your insight. if you hear me well, I hope I wanted to get back to two items that you developed at the beginning of your presentation. So first of all, how to deal with, practices or situation in which the social fabric is completely disrupted. And secondly, how not to oppose compliance versus resistance as being opposed in situations of emergency. Well, I think that for the past year, we have seen both things happening in different contexts. You can take some countries, perhaps with limited resources, which have been hit maybe less by the number of infections or deaths, but much more by the lack of tourism, and therefore bringing into poverty a number of people who were heavily dependent by this. And in more rich societies, like Western countries, think of the United States, but also Western Europe, where the entire way of functioning has been completely altered, not just because people cannot go on weekends flying, from one city to the other, but because everything was closed, no cinemas, no theaters, no museums, no arts, no nothing. So how can we deal with that in a meaningful way? How anthropology can help, if not translate, put things together also for society, for politicians, and for scientists.
Thank you. Thank
when
you
I
very
mean
much for your
scientists, I
question.
mean epidemiologist, virologist. You're also a scientist. Sorry, I was meaning about, of course, life scientist. Sorry.
Yes, thank you. Thank you for your question. I think this is a very pertinent one. I think COVID-19 is, of course, a unique situation in that we do not have, if you want, a kind of a short-term outbreak, but we have something which has already gone for a year for most societies. And it is impacting every aspect of social life. So it is, if you want, a total social fact in anthropological parlance, in the sense that it is challenging not just everyday life, not just work, not just schooling. It is, I think, challenging the very way in which we perceive our relation to the world. And anthropology is uniquely positioned to study these effects. Now, whether this is in a society, say, which has a very violent outbreak with big numbers of deaths, or a society which is very severely affected by draconian lockdowns, let's say, it is important to adopt if you want, a non-functionalist approach. I mean, a functionalist approach would assume that each society is in some sort of self-organized equilibrium, and then you have an epidemic and the state responses to the epidemic which disturb this equilibrium, and that society is striving for rebalancing itself. I think that's a very redundant way of seeing things. All societies are internally antagonistic. They have conflicting interests and agendas, and epidemics, as we know from history, and I have to say that a lot of my work is historical, societies or different parts of society find the opportunity within the unsettlement produced by epidemics to shift patterns of power, to change patterns of kinship, etc., etc. And I don't think this is necessarily something negative. You know, societies are living processes. They're not fossilized, completed processes which have reached some sublime level of organization, which we call parliamentary democracy or whatever. They are dynamic processes that will continue to evolve to unknown destinations, right? And this is something that we need to take seriously, rather than try to push the brake all the time and make societies go back to where they were started. This idea of going back to normality, you know, it is perhaps the most damaging idea in this whole epidemic.
Yeah.
Hi, Christo, thank you for your talk, and I'm very interested about your ideas. So, I mean, regarding the COVID-19, can you hear me well? Christo, can you
hear Leo?
No, sorry. No, it's choppy. I can hear a few words and... Thank you.
Right. Hi, Crystal. I mean, it was an excellent talk. I like your concept. I mean, I'm a bench scientist, so I mean, basically, I mean, you give me a new horizon about this. So it was very interesting. I just want to know, I mean, now, what is the role of this anthropology on, say, on geopolitics, or maybe even for culture? I mean, like, for example, using a mask wearing as an example. You know, in Asian countries, when we heard about SARS or COVID-2, and then many countries in Asia try to wear masks, and it is the citizens. But this actually contradicts the concept or the dogmas in, say, in some Western countries. And actually that create a lot of conflict. I saw people wearing masks in Western countries being hit by some local citizens and so forth. So, I mean, how anthropology can able to actually facilitate or try to reduce this type of a friction or conflict? Thank you.
Thank you very much. Thank you for your question. I think to take the example of masks, what is required is both an ethnographic and a historical approach. We must not see this as an exclusively presentist process. The use of masks in China and East Asia have a century-long history. And if you go back to the first application of anti-epidemic masks in China, which was in Northeast China in 1910, you had equally violent and troubling reactions to this new device. So I think that is what anthropology and history can help us do here, is to say that the acceptance of the mask, say in China or in other parts of East Asia, is not the result of some inherent cultural trait. It is simply the result of a century's use of this protective device, which had its troubles to begin with, but which was adopted by different political regimes and different medical institutions across East Asia for over 100 years. Whereas in the West, it is a newcomer. And of course, when something is new, it's going to create reactions of different sorts. Whereas if you want, in China or in East Asia, the mask is a routinized object. Right? It has become a routine object. In the West, it is still a politically charismatic object, if you want to call it that. It is something which attracts meanings and conflicts. But that doesn't have to do with culture itself as a reified category. It has to do with complex historical processes.
Fred, any question from online?
Yeah, we have two questions online. The first is from Malik Paris. Who says, your point about new type of collaboration is well taken, and who ask if you can give examples of a productive collaboration in an epidemic.
Yes, I can give you a self-serving example of a project I've just started with biologists across Tanzania, Madagascar, and the UK on rodent control for leptospirosis and plague control in Madagascar and Tanzania. And this is a project which aims to integrate ethnographic and epidemiological approaches in ways which is both mutually respective, but which creates new questions. In the sense of our aim is not simply to see, for example, how rural villagers in Tanzania understand rats or how they employ rat controlling devices, or to teach them new ways of rat control. The idea is, through understanding their interaction with the rat, to see how we can optimize different policies and different interactions, which would eventually, I'm giving you a random example because we haven't started the fieldwork yet. If, say, rats were ancestors, in their cosmological system, this would not be debunked through a process that would prove that they are not. It would be taken seriously and integrated in a response to this epidemic risk.
Another question from Harry Wu. He asks, when scientists are using computational models, cell studies, and animal experiments to pinpoint the host of pathogens, how do you see it as an anthropologist? Do you think that that animals are made voiceless in these experiments, and how do they fall into victims of blame, such as bull of bats or pangolins?
Mm-hmm. Yes, thank you, Harry. This is a great question. in a way, this is more a question of STS, of science and technology studies, but anthropology is also interested to see how the agency of animals plays out in experiments or in field studies. And of course, we are concerned, if I can speak for anthropologists, about the stigmatization or demonization of different animals in different epidemic emergency contexts. I've worked a lot on marmots in Mongolia and South Siberia. I'm now working on rats. And it is a very complex social process in which scientists play an important role. For example, the transformation of the rat from a nuisance or a vermin into an epidemic kind of villain, which took place at the beginning of the 20th century, was, of course, if you want, scientifically based, because the rat was discovered to carry plague. But this villainization of the rat also had significant consequences on the ground, which were very destructive, especially for colonized native societies, due to very intrusive measures taken by colonial authorities against the rat, which did not understand its role in local systems. It did not understand the whole complex interspecies and multi-species assemblage that was in place.
So I have a question. So for my understanding, anthropology is always a kind of a prospective study. So you have a question, so you want to understand the behavior of people, then you start up some human experiment and understand what they're thinking, what they do. But in outbreak, so it's always related to the human behavior before these things happens. So how anthropology studies help us to understand the previous human behavior that caused the consequence to the current outbreak?
That's an excellent question. I think Frederic mentioned this as well. I think anthropology is very careful to make the distinction that it's one thing going on the ground after an epidemic has started to study, say, how people interact with bats or with rats or mosquitoes. And it's a completely different thing doing this study outside an emergency framework. And I think the latter is essential. And that's why I know both Frederic and I are very much promoting studies, both PhD and postdoctoral studies of these long-term relations with potential zoonotic hosts in different geographical and ethnographic settings. Because the problem is that once an emergency settles in, as Frederic said, then all these perceptions get distorted and changed. So what you're studying, if you go on the ground now in a setting where the pangolin is being suspect as an intermediary host, you're not going to get the data and the information you would have gotten if you were there in 2018, right? You're going to get completely different information. Both studies are very important, and if possible, we should be doing both studies, so the before and after, and then we would understand very important things. The problem, of course, with conducting ethnography on the ground during an emergency is ethical and practical, and sometimes it's not possible to reach these locations. And the problem, to be very frank with doing ethnographies of animal-human relations focusing on zoonosis before an emergency is that usually funding bodies find it rather unattractive. So we have a problem with funding structures in the UK and in Europe where emergencies are being funded, but long-term processes are not. I'm lucky, and Frederic is also lucky to have overcome this obstacle and have established long-term research like this. And I think this is becoming now a very respected field of study, at least in the UK and France. And funding bodies are seeing the benefit and the point in this. And this is leading to a very important corpus of work on zoonotic diseases in different contexts, which are not necessarily emergency contexts. And this is something that has immense value, I think.
But one of the key questions is how do we avoid bias from some of the behavior that has been done for many years? And then suddenly there's an outbreak happened and we look back, oh, that's just the behavior that caused the outbreak. One of the excellent example is the avian influenza. Before 1997, when people, in the market, just in my residential area, so we blow the eggs of the chicken to see whether it's healthy or not. No people complain about that. And after 1997, we know this is a risk of getting infection. So how do we look back when at that time that this behavior is kind of association?
So do you mean how do we understand behaviors before the emergence of this?
Yeah, without bias, yeah.
Well, I mean, that would be something for a historian or for a historical ethnography, and really depends, well, on existing archives or oral histories. But of course, oral histories will have, again, the problem of the memories being filtered through the recent emergency. Ideally, that would be archival work, but that may or may not be feasible depending on the context.
Any question from this day, from the theater? Yeah, Roberto?
Another one. I want to have, again, your comments, maybe even Frederick, of course, and anybody can comment on that. You mentioned in the case of, for example, criminalization of animals, the role of colonial power or any type of power remote from the cultural roots of the place where this power was exerting its authority. I would like to be provocative and try to draw a parallel between perhaps also a community of scientists who are obviously, if not enhancing what they're doing now because of the pandemic, and even of politicians or people with authority that are gauging their own actions regardless of what happens on the ground. That is, we're talking, for example, restaurants or bars or nightclubs or you name it, which are also rooted in the social fabric of different countries. And this split, this dichotomy, is more or less obvious depending on how vocal people are in those countries. But they do exist. Sometimes I think that, most of us, our salary is not affected by all the measures that have been proposed. But for many, many people, this has had an incredible impact on their lives. Malik Pieris mentioned yesterday 80 million more poor in 1 year. I read this morning 2 million just in Kenya. Yes, as an anthropologist, as a human, what do you think?
No, I agree. This is a huge challenge. I don't know if it is feasible if we have enough anthropologists to study COVID-19 and its impact in different societies. Medical anthropology is a subdiscipline, or anthropology of medicine is a subdiscipline, and anthropology of epidemics, it's maybe a dozen of us around the world or so. It was never a big thing in anthropology, right? So we were eccentricities, if you want, but which proved to be, very much needed in this context. But I think that what is important is precisely to combine different disciplinary approaches. For me, combining historical approaches and ethnographic approaches in the study of epidemics is key, because only through the historical study can we see the genealogy and the formation of different concepts and different human framings of processes of infection, but also of epidemic control, right? To draw very nuanced and fine histories of epidemic control measures, their material aspects, their technological aspects, is essential in this context. And the mask is one example, but there are many others that both historians and anthropologists have been looking at in this past year.
Any more questions? Any more from online?
No more questions, but I wanted to thank the HKU-Pasteur Research Center as a place of collaboration between anthropologists and biologists. And I also wanted to thank Mathilde, who has been very helpful in all the making of this event, at least this panel.
Yeah. So, Sophie, you have one more? Oh, yeah. Oh. He left. Yeah.
It feels like such a coincidence with the COVID-19 pandemic being 100 years from the 1918, influenza, pandemic. And some of the images that we see are so familiar to what they're recommending now for public health measures, like even outdoor schools and the mask wearing and a lot of the public health messages in newspapers. And it feels like within 100 years of how much research and science has been done to sort of still be back in the same place that we were 100 years ago, what have we really learned and changed? Or is it just that these are fundamental human nature things that we need to control pandemics? So I just think there's so many similarities in the history books of the 1918 pandemic and this COVID-19 pandemic that it feels too coincidental. So is this like a cyclic human behavior, or is it just that that's the only tools we really have, that we're going to go back to the same things like the plague masks that you sort of see from Venice, and that we have the same masks in 1918 and masks now, that we have really limited tools that we've had throughout history? It's not so much of a question, but what do you think?
So was this a question to me?
Yeah, so the 1918
Yeah, okay.
pandemic looks
Sorry, I didn't hear you very well, but I think I caught the main line. Yeah, I'm rather reluctant to adopt a linear perspective on history as a process of progress. I think that this is yes, it's an interesting notion of the Enlightenment, but I think it lacks historical evidence. And definitely the idea that we learn from history, I don't think there is much evidence from that either. I think that's, again, an interesting framework of the last century or so. But so I wouldn't say even that the mask that people used in Manchuria in 1910 or in, say, the United States in 1918 is the same object, right? I mean, it has the same materiality and the same molecules, and it looks the same, to an AI machine, perhaps. But that's precisely why AI is not intelligent, because it cannot see the symbolic fabric of this mask and the social relations behind the mask. The mask is a social object. It's not just a composition of fabric and elastic bands. So I wouldn't say that we are going back to anything. This is not possible, in fact, as a social process. And of course, I think we delight in our societies to find comparisons with the past, to say, oh, look, the mask back in 1910, or look at the quarantine in Frioul in 1901, or in Sydney. But this is just a kind of, well, it's not just, it's an important part of creating identity and creating meaning in our societies. It doesn't mean that this process is in and of itself historically meaningful. What I mean is that creating these connections and these remembrances are important for us so that we cope with the epidemic. Now, when it comes to an actual historical scientific examination of these processes as similarities and differences, it's a completely different question, right?
Yeah. So thank you, Crystal. And also I would like to thank all the speakers in these sessions. And may I invite our co-director, Roberto Busson, to give a closing remark?
Okay. Thank you. No, yeah, I mean, no, I'm prepared in the sense that I wanted not just to thank the speakers and the panelists and the discussants for the past 2 days, and the organizers. Everybody has already said that, but also try to think back at some of the issues that have been raised throughout these 2 days. And one thing that I would like to underscore is that despite all the difficulties in working during peacetime, I don't like this is the term, but say between pandemics, and despite the difficulties in creating the tools necessary or useful to control or to control in more effective fashion outbreaks, one can say that in this particular case, there were several alarm bells that rang quite early, and that in a way or another, we were able to follow this pandemic in real time. You know, again, there was a typical pneumonia at the beginning, not understanding whether it was really coming from one way or another. And then after a while, clearly understanding that it was quite serious, a country the size of China getting into massive lockdown of a province that at least has the same inhabitants as probably France. And then everything proceeding fast from one country to the other, but at the same time trying to really deploy all the different tools, all the different heads-on that had been developed over the past decades, say, in order to have a better control. Now, one can say that this perhaps may not have been the case, but if one considers what could have been the consequence if nothing had been done, if there had been a complete reaction to this pandemic, it is clear that both from the scientific, perhaps social, epidemiological, and even the anthropological and social, social science aspect, there has been a very different reaction now, and this is why, I mean, it's not, history is not a linear process, and this is why we cannot draw parallels, but this new society or this different society that had global society that had evolved over the past 20 years thanks to traveling, communication, and many other factors, had a different way of reacting, I think, to this pandemic. We are still in the, I don't know whether in the middle of it, towards the end of it, depends how you see things, but whether it lasts for 6 more weeks or 6 more months or 6 more years, it will end. And this is something for which we can be sure. And whatever happens next, it may not be necessarily going back to normal. And I may agree that with Christos, there's a concept perhaps is not a very interesting one. But on the other hand, we are also humans, and a very different type of, perhaps, of animal species. And I think that animal behavior is also part of the way we behave. Even if we are thinking about disease, we are thinking about ways of protecting us from disease, we still have our own way of doing things. And this is part of the effort that we need to make in order to get a better control of it. My other thought would be in particular to the problems of access to health that have been magnified during this pandemic. Not just in countries where this access to health has been unequal for maybe forever or certainly for many, many years, but even in countries where this access to health should be possible for everybody. And clearly it has been shown that the way that the health system had been managed for the past decades, in particular in many wealthy countries, with, I think, almost with an eye only on profit and cost-cutting measures, is something that has had dramatic, catastrophic consequences during the past years. And the final remark is that it's a sobering one, perhaps. I think that a lot of things have happened of recent, but it is really the first time that a dangerous pathogen has been a permanent resident in countries that were no longer used to have this type of threat. There are very dangerous pathogens that are still quite resident in many countries. We talk about Ebola. There are new outbreaks again, but this level of threat was obviously no longer perhaps thought of or tolerated in many societies. And I think that we need to understand that in a world where the human, animal, and environmental interface is still expanding because the humans are increasing in numbers, because the possibility of venturing into environment of different kinds is really expanding, where animals are really closer to us because of societal, cultural practices, industrial practices, etc. These encounters will definitely not decrease. We need to be ready to do what we can, and in particular to have perhaps swifter actions in the future. But again, because of the social problems, it's not very easy to say, I don't know, put the whole world at standstill. Can you imagine if on January 22nd of 2020, the whole world had convened in a meeting and said, okay, now we close everything for the past 4 months? What would have been the reaction, I think? And how could one have persuaded 7 billion people to go into a world lockdown, saying that, say, 3 months, we can make a model. 3 months of lockdown at that time would have basically dissipated or vanished any subsequent danger. And therefore, you can make them the cost benefit, how many trillions would have been saved, and so forth and so on. So basically, all these rambling at the end of these 2 days is that to say that the various disciplines that we have tried to integrate into this colloquium raises so many questions that we are unable still to integrate, that we want to do more in terms of collaboration, and it's great that there were many examples being shown. I think in particular also at the countrywide program of Cambodia, illustrated by Véronique Chevalier. But we need to also remain a little bit humble about what we can really do and what we can really apprehend. Otherwise, it's much better than we keep doing our work. And hopefully we can see each other in person for another meeting on another topic in a different context, but with the same enthusiasm, with the same desire for enhancing our knowledge and our mutual understanding. So thank you to everybody.